Okay. So I feel like a little bit of a fraud here 'cause I'm a molecular anthropologist, and most of the work I do actually involves understanding human health in the Pacific, and particularly metabolic disease. But it turns out that to understand this, we really need to understand more about how people distributed across the Pacific, the relationships they had with other people, and sometimes that involves looking at the animals and other critters that they carry along with them. And this actually also suits me quite a lot because I really love chickens. And so this has allowed me to combine the work I love doing at work with something I love personally. So we'll take you on a journey looking at chickens in the Pacific. So when you look at archaeological sites throughout the Pacific, chickens are actually very broadly distributed. They seem to have arrived very early on, and you see them in some of the quite early Lapita sites up here in Musel, and up in Wattam. And they also distributed down with the Lapita people down to Vanuatu very early on. We see them down in Tonga around this time too. So they were part of the early kind of transportable landscape that we kind of talk about quite often when we're talking about Lapita, but we also see them a lot in later sites as well. They disperse quite rapidly out throughout all of remote Oceania, or mostly, apart from down here to New Zealand. So you do have this distribution of domestic animals across Polynesia, where you do have some of these domesticates that we think were transported missing, like pigs, which we don't see in certain islands. We do usually see chickens, and we see chickens very abundantly. So it seems like they're filling an important niche where protein is needed. So, yes. So this falls into the commensal model for tracking human mobility in the Pacific, which Catherine talked to you a little bit about yesterday, where we can use the genetics of these animals that were carried along throughout the Pacific by people. Obviously, chickens can't row a canoe and can't get to places by themselves. So we can look at the DNA of these animals to look at interactions between people in the past. We'll take you through a quick journey, a personal journey of chicken variation in the Pacific. You can see, obviously, we've got a lot of chickens in Vanuatu. They're quite phenotypically different these days. Lots of different chickens, very pretty, A+. We also see them, I've also obviously been to Rapa Nui. Again, we see chickens throughout Rapa Nui. And here, Rapa Nui people are obviously very close to my heart because chickens here were clearly quite important prehistorically. And you see the construction of these haere moa, or houses for chickens, in the past. And we can see some contemporary chickens here using these haere moa. And we've also got some quite cool dendroglyphs as well, where you can see here they have carved a few chickens on this piece of rock, and I think this is a haere moa at the top here. So the fact that people have gone to the effort of carving this into a rock clearly suggests that chickens are quite important. And again, we've got local chickens here as well. Here are some I met the other day. Very cool. Love them. Anyway, why should we study chickens? I've kind of covered this a little bit already. They're part of this transported landscape taken around the Pacific, not only by the Lapita, but people moving out into Polynesia more recently. Chickens can't move themselves between islands, so they rely on people to do this. So it can tell us more about people's movements and their social connections in the past. It can also contribute to understanding of domestication processes more globally. So the process of getting wild animals and turning them into things that are a bit more useful to us so that we can grow and travel more easily. The problem is that modern chickens are, well, they do a lot of breeding with each other. So we've got a lot of phenotypic variation in contemporary Pacific chickens, but we don't necessarily know whether this is something that if we went back a few hundred years, this would still be the case. And this is because we've got the early introduced chickens, but we've also got chickens that were introduced by Europeans, and these guys do not interbreed. I mean, chickens, they make babies all the time. Anyway, so this makes it quite difficult because we can't really look at modern chickens to untangle what was going on in the past because we've got all this admixture, just like we see with humans, with chickens introduced in the last couple of hundred years. So this means that for taking a commensal approach, we really need these ancient chickens and the bones of these ancient chickens to understand paths of migration and into island contacts and things. So this has, to a certain extent, been done. There have been a number of ancient DNA studies which have generated partial mitogenomes, so a couple of hundred base pairs of ancient DNA, to look at this specific question. And what Story et al. Found, that working with Lisa about a decade ago or so, was that it looked like there were a couple of distinct chicken lineages, which they were calling lineage D and lineage E, that were distributed out across the Pacific. And these were linked to different temporal introductions. And so it seemed that we had a relatively early introduction of haplotype E, and then a secondary later introduction of haplotype D, which comes in a little bit later. And this is kind of cool because it tells us a lot about connections with people. It also, around the same time, some chicken bone was found in the site of El Arenal off the coast of Chile. These came back with a nice, secure archaeological context with radiocarbon dates that dated pre-Columbian, so it definitely looked like these guys were introduced by probably Pacific voyagers coming along the coast of Chile, probably picking up their kumara and things along the way, and being left behind. The DNA sequences of these chickens matched the ones that were being found in East Polynesia, so belong to that what we were calling haplogroup E. And so it seemed like a really nice story of Pacific chickens being introduced to South America. However, these results did become contested back in 2014 by a group from ACAD, who suggested that all of the haplogroup E chickens were actually a result of contamination, and that all of the Pacific chickens in the ancient times belonged to haplogroup D. And so this is actually cast a really long shadow over Pacific genetic studies, and it's had quite a lot of repercussions in other studies that have happened since. But quite interestingly, a Chinese group, Miao et al., they've actually found in their study of hypervariable, small hypervariable regions of DNA, that you can't actually tell the differences between haplogroup D and E1 based on that particular length of DNA, based on the mutations that you see in there. So while we now know that they were right, and that Pacific chickens do all belong to haplogroup D, it doesn't necessarily exclude the possibility of two subtypes of haplogroup D within the Pacific. It also does suggest that we should be all transitioning to technology that is capable of detecting contamination, and so we'll cover that in a second. So what we have done is move on to sequencing a larger portion of the DNA. So you can see here in this figure that we've got the control region. This is what the old studies were looking at. It's only a few hundred base pairs. But for our study, we have now sequenced the whole mitochondrial DNA. So that's the maternally inherited 16,500 individual letters inherited through the mother's line. So this gives us a lot more resolution. It allows us to figure out a lot more specific lineages. And also the technology for doing this is a bit different. So we were not, obviously, convinced by this contamination argument for the previous results. So we have used the latest technology to check this out. This is the next generation sequencing. Next generation sequencing has a lot of advantages over the old Sanger sequencing methods, which was the technology that was used in both of the previous studies I was talking about. And one of these is that it sequences all of the DNA molecules that are present within the sequencing library. So this includes the DNA from the chicken, it includes DNA from the environment, and it also would involve any sequences that are coming from other contaminating sources, such as reagents. From the experimenters like me, we can see them, they're all there. And so this means that if there was DNA from chickens contaminating our ancient chickens, we would be able to see that within the data that we get back. So we have prepared DNA from 54 chicken bones throughout the Pacific, including New Caledonia, Tonga, Samoa, Hawaii, where else? The Marquesas, as well as a historic site of the Isla Mucha off the coast of South America. We have successfully sequenced only 12 ancient Pacific mitogenomes and 2 historic South American sequences from these chickens. And I will note that generally ancient chicken DNA from the Pacific is very poor, which is why we've got such a poor success rate. Compared to some of the other animals, including kiore, like Catherine talked about yesterday, the recovery of DNA from chickens is pretty sad. Anyway, these are the results we've got. I've just made it into a network which shows the relationships between different samples. You can see that some of these, so the colored dots are different individuals that have been sequenced. Some of these, like these ones up here, are ones that have previously been sequenced by other labs and which are available in online databases like GenBank, whereas our new results are down in this area down here. So you can see that all of our our Pacific chickens, which are these green ones and these dark greeny ones here and the pink ones, they all belong to haplogroup D1. And they cluster broadly into two groups. So we've got what I'm just calling group 1, which is over here, which includes some ancient chickens from Rapa Nui, some ancient chickens from Hawaii, some modern Marquesan and Fijian chickens, and a few modern New Caledonian Vanuatu chickens. They're clustering this group 1. And we've got this other second group over here, which I'm going to call group 2, which is separated by about 10 individual mutations, making it a quite distinct group. And this includes a few, well, we've got only one Tongan chicken here, which dates to the Plainware period, but we have a few slightly less good quality, but definitely very similar to this specific chicken, samples coming from 3 different Lepida sites in Tonga. We've also got these 2 chickens here, which are from Micronesia, so from Yak and Chook. They're also quite similar, and they're in this this group 2 group. And we've also got a few Hawaiians here, so I think it's 3 individuals. So it does suggest that we do have two chicken lineages, which reinforces the work that was done by Alice and others back in the earlier 2000s. And they are differentiated by a number of variants, so that these are chickens that were brought along to where they are pretty much as they are. It looks like we've got some sort of temporal control here, where we've got these early Lepida and Tongan plainware chickens sitting in this group 2 group, compared to these predominantly East Polynesian chickens over here. So that's quite interesting. And to just reassure you, we can definitely tell the difference between D and E chickens using these whole mitogenomes, because we've got these historic samples from Isla Mucha, so in Chile, sitting up here in a completely different part of the network, showing, we can definitely tell the difference between E chickens and D chickens. So it's not contamination. So just re-sum that up. We do have all ancient Pacific chickens belonging to haplogroup D. And if we go back here, we've got a probable origins. Haplogroup D arises up in the Philippines and the island Southeast Asia area. So the Pacific chickens that we're seeing in archaeological sites probably arise in island Southeast Asia, which is what we know about chicken domestication anyway. We have got 2 distinct lineages separated by about 10 mutations, and we can definitely tell the difference between E1 lineage chickens and D lineage chickens. And we can confirm, having looked very closely at all of our samples, as well as negative controls that were prepared alongside the samples, that we have no contamination within these samples. So we're pretty certain that what we're seeing is right. We've also reinvestigated the South American connections question. Sorry. Yeah. We've unfortunately not been able to get the original bones for that LRNL result that we talked about earlier. They've not been allowed to come back to us for retesting, but we've been sent some recently excavated bird bone from new excavations at LRNL to try and confirm whether or not the original findings were legitimate. Unfortunately, the bones that were sent to us were not morphologically identifiable as chicken. There were no diagnostic portions left on them. We ran them anyway, and unfortunately, they're not chicken. The specimens belong to a great tinamou, which is a small galliform endemic to South America, but they had very, very good DNA, and they had very good depth of coverage, and they were also clean of chicken contamination. So we're pretty confident that if we were able to get the bones back, we would hopefully be able to get a result. And tinamou and chickens, this is just an alignment of the sequences. are very, very different. So you can see at the top here, I've got the chicken reference sequence. We've got the 3 original chicken sequences from El Arenal that were online. We've got the reference sequence of a Chilean tinamou, and we've got the 3 bones that we sequenced last year. And you can see that they are very, very different. You cannot confuse a chicken with a tinamou. So fingers crossed one day we can get those chicken bones back. We've also, so what's next? We've sequenced some genome-wide data from 4 of our ancient Pacific chickens, including the Lapita Tongan plainware chicken I talked about earlier, a couple of ancient Hawaiians, and one of the ancient Yapese chickens. These are still undergoing analysis, so we'll hopefully be able to tell more stories about what was going on soon. And just a note, as I've said multiple times, I really love chickens. They're my absolute favourite. They're fluffy, cute, lay tasty eggs, like keeping them as pets, like studying them. So if anyone has some nice archaeological chicken bones lying around that they want analysed, I would be very happy to receive and analyse your chickens for you. Anyway, that's my talk. Mahururu. Thank you.